Good afternoon, everyone. Today I'll be presenting about our work in microbiome and its implications in cancer. I'm Pratik Chandrani, computational biologist from Tata Memorial Center, Mumbai. As we all know, our body is made up of microbial cells, which are in numerical sense far more than human cells in our body, and almost every part of our body has some cells which are involved in various functions, and those functions are absolutely critical. And without this microbe, we cannot survive absolutely. But how do we study these microbes? There are many different techniques available which are specialized for microbial sequencing, but they require add-on experimentation and analysis. Our lab usually focus on human sequencing, so we hypothesize why can't we integrate one single sequencing assay wherein we study human DNA as well as microbial DNA and derive the interpretation. So how we actually do is from a human sequencing assay, we get the data relevant for human analysis, and that when we align to the human genome, as I have shown here, chromosome 1 and 5, we get to see point mutations, indels, copy number variation, amplification, as well as fusion. And then whatever is not mapping to human genome and whatever is abundantly present in the tumor site is also captured in the sequencing assay because of the hybridization probes that we use. Those are not 100% specific. So this we identify as a microbial origin of DNA, and we have prepared a database of a few million microbes genomic DNA. By analyzing against them, we can identify which bacteria or which virus they are coming from. So by using this concept, my post study when I was a PhD student in Dr. Amit Dutt's laboratory, we developed HPV detector to identify HPV from cervical cancer whole exome sequencing dataset. And for that, we at that time we prepared 140 HPV strains in the database, but now it has been extended further to contain at least 190 different HPV strains. From this analysis, we identified that HPV 16 and 18 were the most predominant, of course, as expected, but we also observed several other HPV strains which were not otherwise known to be predominantly observed in the human samples. Now, if you pay attention here, some of these strains have much higher proportion compared to average. But if you ignore that we 16 and 18 ratio, as I'm showing here, so HP 16 and 18, if we don't look at their relative quantity, but rather look at their binary status, we see that in majority of them, 16 is present, but along with that, we see at least 3 or 4 other HPV strains present. And few of these strains are not known to be associated with carcinogenesis. However, when we do clinical correlation of these patient samples, the one which are having only single strain of HPV, they have usually performed much better in the clinic, but the ones which are having multiple strains infected among them, they were performing much worse in the outcome. So this study was published in British Journal of Cancer, and since then more than 200 different users across about 50 countries are actively using HP detector. We ourselves have followed up further with gallbladder cancer analysis, which is perhaps of more importance in this region, wherein we identified non-typhoidal salmonella infection to be clinically associated. And now many and many studies are coming up where a non-typhoidal salmonella diagnostic tests are also being introduced. The take-home message that I'm trying to convey here is that metagenomic approach from a human oriented sequencing assay, having genome-wide approach, can be reused for microbial organism study. Now, to extend this further, we have developed HP detector into version 2 of the computational tool, which we call now infectious pathogen detector. And one of my junior, Sanket Desai, took this work further up, where we identified the landscape of microbial infections across various different cancer types. So on this panel, we have breast cancer here, followed by panel 2 of cervical cancer. This entire panel is colorectal cancer, and these are all head and neck cancers. These are all sequenced at Tata Memorial in-house. Now here we see HPV and several other expected organisms to be identified in cervical cancer, oral cancer, and so and so. But to surprise to us, we observed that Fusobacterium was highly enriched in several oral cancer cases, similar to colorectal cancer cases. So when we were doing this study, Fusobacterium was reported to be oncogenic in colorectal cancer, but it was not reported ever in oral cancer. So this is our one of the first report on that line, and we further show that in the patient samples recruited from Tata Memorial, the Fusobacterium positive patients have much worse survival compared to Fusobacterium absence or very low level, and the same was further confirmed with even larger cohort of patient samples from USA. We also take another step wherein one more PhD student from our lab, Vaisakhi, who is working under the guidance of Dr. Kumar Pravas, and in collaboration with Dr. Anuradha and Dr. Amit Dutt, identifies Cutibacterium in thyroid cancer. Thyroid cancer, to our understanding, is a more sterile organ compared to other body sites. But to surprise to us, we observed that Cutibacterium is highly predominant in thyroid cancer, and not only predominant, it is associated with immunosuppressive genotypes and biomarkers in the thyroid cancer patient samples, and anti-inflammatory response generating biomarkers were also present and enriched in Cutibacterium positive cases, and their overall survival was statistically significantly lower. So from these three studies, we have seen how individual pathogen-based identification can be useful in various clinical aspects. In all of these essay, we have used the same type of microbiome analysis approach. We take it further by one more step in the next study, where we identify that a red oncogene in a medullary thyroid cancer is directly affiliated or associated with dysbiosis in the microbiome, which is to our understanding first of its kind observation we have made here. So here what I'm showing is a landscape of medullary thyroid cancer in which red is predominant. Roughly about 49% of Indian medullary thyroid cancer patients are positive for rate, which is very similar to the global data. Now, among these, when we compare the entire downstream mutation landscape, we observe that rate mutant patients are majorly mutated by long-term potentiation genes and genes involved in various infection and thyroid signaling. And rate wild-type patients have majority of the mutation accumulation in transcriptional dysregulation. They are also, well known to be associated with poor survival. Interestingly, when we compare their overall microbial diversity, we see that there is not significant difference in the microbial diversity, although numerically they do look different. But when we do individual species-wise enrichment, we observe that there are several organisms which are highly significantly depleted in red negative patients, and the other group of species which are highly enriched in the red positive patients. So the same type of organism I'm showing here numerically, these all patients are red positive, and these all patients are red negative. And if you see the relative quantity of each of the microbial species, you will see two distinct group coming out here. All of these patients are having relatively higher quantity of these microorganisms, and all of these patients have relatively higher quantity of these microorganisms. So this is what we call dysbiosis happening in the context of a red mutation. And we are further conducting experiments in the lab to understand how human oncogene is associated with microbial dysbiosis, and whether we can exclude this for the diagnostic, prognostic, or therapeutic applications. Now, we have seen how a tissue microbiome is relevant for cancer study, but microbes are not only playing role at the site where they are found. One of the best example is gut microbiome, wherein bacteria in our gut are known to induce several signaling pathways in the body, and they are known to control the immunity of the individuals. And there are several biomarkers and several pathways which have been very well established in this line of work. A few of them are directly affiliated to pathways which induce the immune potentiation, and some of them are directly known to inhibit the immunogenic activity. Now the same can be exploited for immune checkpoint inhibitors, and that I'm going to talk how a diagnostic biomarker can be derived from the same. Now few of these gut microbes are known to either have some metabolites which are interfering with our signaling network, or they have some toxins which are inherently produced by the bacteria, and those toxins are inducing DNA damage and reactive oxygen species induction, which further results into the pathways which are relevant to cancer. But either way, either route they take, overall their dysbiosis results into tumorigenesis, and it has been very well established in gut as well as several other cancer types. Now, how do we use this information in clinical settings? So here, one of the data I'm showing is grouping microbes into two different categories. One are called firmicutes, which are known to be part of healthy gut, and they have been identified by systematically analyzing non-cancerous and no any other disease condition harboring healthy population. And other type of organisms are called bacteroidetes, which are actually the ones which are acquired because of different lifestyle or food habits, and they are not supposed to be part of human gut. If we can quantify each of these bacteria which are falling into this category and each of these bacteria, and if we take the ratio, it is usually in the positive, the ratio of firmicutes divided by bacteroidetes. But in a cancer cell, particularly when you want to infer response to immune checkpoint inhibitor, people have shown that this ratio is directly associated with it. This ratio, if it goes towards zero or in the negative sense, then there will be absolutely no response to immune checkpoint inhibitors. If this ratio is higher in the positive numbers, usually towards 5 to 10 or higher than that, then that indicator of a very healthy gut, and there will be very good immune checkpoint inhibitor response. So this calculation is being implemented in my lab for these computational tools that we have developed, and once we validate this, we'll have more in-house data available for the same. So overall, what I have presented is our journey of microbiome analysis, in which we started with rediscovery of HPV by using this metagenomic approach, which we have taken further to non-typhoidal salmonella and fusobacterium infection and cutibacterium infection detection in various cancer types. Furthermore, what I have also shown is expanded metagenomic approach to identify dysbiosis of overall microbiome, wherein we have discovered that rate mutation also drives such kind of dysbiosis, and more further functional studies are being conducted. This study is already communicated, and I hope it will be available in the public domain soon. Furthermore, we are taking this approach one more step to use it as a diagnostic biomarker by taking a clinically usable ratio to inform which patients are more likely to benefit from immune checkpoint inhibitors. And given that diversity of lifestyle, food habits, and overall genetic subpopulation in India is enormously huge, we propose that we should do more systematic microbiome profiling in almost all the part of our country. And TMH Varanasi has also shown great interest, and we are starting a collaborative study to do such microbiome profiling here. And I'll be also very open to other clinical centers whosoever can contribute gut biopsy samples for microbiome profiling. Will be very open to collaborate and take it further. So that's about my work. This is my laboratory, and all of our members are, this is what I call extended family, and not the professional network. And we have postdoc, PhD, and JRA positions available in the lab this year. So if you know anyone who might be interested in this kind of approach, please do connect them with us. I would like to thank organizers, particularly Dr. Mishra and Dr. Akhil, for providing me this opportunity, and